Okay.
Can you have your you're muted.
Okay, can you hear me? I'm sorry, I cannot hear you.
Oh,
I
right,
think
so you're
I'm still
speaker.
muted.
I'm sorry.
Yes,
Okay.
yes. Okay, please, Professor Chang, the floor is yours, so you can start your presentation.
Okay, thank you very much. Okay. Good afternoon, everyone. My name is Mi-Sook Chang at the Seoul National University. And thank you very much for having me as a speaker in this prestigious meeting. This is my great pleasure and honor to be here. And the title of my talk, Glial-like cells from bone marrow-derived human mesenchymal stem cells protect against ischemic stroke through insulin-like growth factor binding protein 4. I changed the title a little bit from the brochure. Hold on, please. Okay. So stroke is caused by disruption of blood flow in the brain, and it results in the reduction of oxygen and nutrients, and leading to death of the brain tissue. There are 3 types of stroke, and I'm going to talk about ischemic stroke today. And purpose of my study is to develop the strategy to induce human mesenchymal stem cells into the functionally differentiated cells with the desired cell fate in the nervous system, especially astrocytes in this talk, and to integrate the strategy of microenvironment modulation and cell replacement to enhance neural regeneration in the injured nervous system. And the ultimate goal is to provide a fundamental basis of the efficient and safe cell therapy for neural repair and regeneration. Mesenchymal stem cells, as you know, they are capable of self-renewal, and they are multipotent, not only into multiple mesodermal cells, but also into ectodermal cells like neurons and glia cells. Because a subpopulation of MSCs, especially bone marrow-derived MSCs, are originated from neuroclastic stem cells. These prominent papers were published in Cells in 2007 and 2008 to prove this. And autologous treatment is possible, so we can avoid both ethical and immunological problems. There are 2 main concepts using stem cells for the regenerative medicine. One is make stem cells differentiated into specific cells to replace lost or damaged cells. But this is minor in most situations. The major benefits using stem cells for treatment is from the microenvironment modulation in injured tissues by secretion of cytokines and growth factors, and anti-inflammatory effects, anti-immune reactions, and stimulation of tissue stem cells and or progenitors. So I differentiated HMSCs into specific cells to replace damaged or lost cells, and also I try to create a favorable environment for regeneration by protecting endogenous cells and by enhancing endogenous repair capacity. So this is a scheme. So this self-renewal HMSCs can be differentiated into glial-like cells to have both microenvironment modulation and cell replacement to potentiate regeneration. And also our lab actually induced HMSCs into neurons and neural stem cells. But today I'll focus on the glia-like cells induced from human mesenchymal stem cells. And then I decided to transdifferentiate human mesenchymal stem cells into astrocytes because astrocyte is required for neuronal survival and function. And these are the neuroprotective effect of astrocyte and antioxidative effect and clearance of glutamate toxicity and anti-inflammatory effect and synthesis and release of neurotrophic factors. And astrocytes also can help oligodendrocyte with myelination and angiogenesis. And I'm going to focus on anti-inflammatory effect and synthesis and release of neurotrophic factors in this talk. The paper is published from Ben Barros at Stanford in 2017 in Nature. There are two different types of reactive astrocytes induced by neuroinflammation and ischemia. One is A1 astrocytes, and the other is A2 astrocytes. This is still controversial, but I will talk about the A1 and A2 type here. And A1 type astrocytes, they are neurotoxic and cause death of neurons and oligodendrocytes. But A2 type are astrocytes, they are protective and to increase neuronal survival and tissue repair. So I thought if I can have more A2 type of astrocytes, and then it will be we can use these A2 type of astrocytes treating neurodegenerative disease. And so I decided to transdifferentiate HMSCs into glia cells, and then we found out that using a peptide growth factor array, and we found that these factors are increased tremendously in induced HMSCs. And also, I looked at the effects of these cells in a spinal cord injury model and also a mouse model of a hind limb ischemia. And this is the scheme of GHMSC induction and cryopreservation. So we optimized the protocol and established the method to cryopreserve GHMSCs in the middle of induction. So we can use whenever we want to use. And also GHMSCs exhibited an elongated bipolar shape glia-like morphology. And also GHMSCs expressed astrocyte glia marker, especially astrocyte marker, GFAP, and S100 beta, and also GLAST. And so these HMSCs have glia-like features. And this is SC means spinal cord. It's a red spinal cord, organotypic red spinal cord slice culture. And so this red filament indicates neurofilament, grows from the spinal cord, slice culture. And LPC is lysolectin, is a demyelinating reagent. So it demyelates and disrupts neurofilament. But if we transplant uninduced HMSCs, actually it increased neurofilament, neuronerve regeneration. But our induced HMSCs, GHMSCs increased more, stimulate nerve regeneration in injured spinal cord injuries. But this effect can be blocked by using blocking antibodies against anti-HGF, anti-VEGF here. However, we found out that I'm going to briefly explain these figures. But these anti-blocking antibodies could not completely abolish the protective effect of by GHMSCs. So we thought there must be some other factors involved in neuronal survival rather than VEGF and HGF. And later, we used hind limb ischemia model to look at the tissue regenerative capacity of these MSCs. And we looked at the primed HMSCs significantly increased blood flow and limb salvage in ischemic hind limbs. This work is done by a collaboration with Dr. Hyus Kim and Seoul National University School of Medicine. So this, and also we looked at the tissue and induced HMSCs enhanced angiogenesis in ischemic limbs, and also induced HMSCs survived better in ischemic limbs compared to control on induced HMSCs. And then, so I thought, so our induced HMSCs may be very efficient to treat ischemia. And so I thought, how about brain ischemia? So I decided to look at the neuroprotective effects of GHMSCs, glide-like HMSCs in brain ischemia models. So we looked at the peptide array result, and HGF and VGF, actually the amount of HGF and VGF from induced HMSCs are much higher compared to those of uninduced HMSCs. And also IGF-BP4 have significantly increased the levels of in our cells compared to uninduced cells. So IGF-BP4 actually binds to free IGF ligands to stabilize IGF. And the IGF and IGF-BP4 complex is cleaved by PAPPA near the target cells to increase the local local concentration of IGF. So we decide to in vitro assays and in vivo assays to look at the neuroprotective effect of IGF-BP4 in brain ischemia. In the in vitro assays, we used primary cortical neurons, and we gave oxygen glucose deprivation damage in primary cortical neurons for in vitro assays, and we also adapted MCAO functional recovery model. MCAO stands for middle cerebral artery occlusion model. That's the in vivo model of acute stroke, ischemic stroke. And this is the result in the in vitro assay. This is red primary cortical neurons. We gave oxygen glucose deprivation for 3 hours, and we treated CM from GHMSCs. CM stands for conditioned medium. And 3 hours later of oxygen treatment. And we found out that this is OGD actually reduced the cell viability up to 50%, but GHMSC significantly enhanced the cell survival of OGD treated cortical neurons. And this is the relative gene expression of IGF-BP4 from GHMSCs. This increases up to 3-fold compared to HMSC. And this is ELISA assay. IGF-BP4 is released from GHMSC up to 8,000 picogram per ml. It's approximately 8 nanogram per ml. So there is a human recombinant protein commercially available. So we looked at the 200 nanogram per ml concentration of recombinant human IGF-BP4 actually produced similar neuroprotective effects compared to GHMSC. So here, so that means, but we have a much smaller, much lower concentration. It's 8 nanogram per ml compared to 200 nanogram per ml. because as you know that the proteins secreted from the cell have three post-translational modification and also proper three-dimensional structure, because they have a more, they are much more stable compared to recombinant protein, recombinant peptide. And also we looked at because since IGF-BP4 is involved in activation of IGF-1 receptor, and IGF receptor have two downstream pathways, or one is PI3-AKT pathway and the other is ERK pathway. So we decided to look at which pathways is involved in IGF-BP4 mediated neuroprotection. So we used PPP. PPP is picropodophyllin. This is IGF-1 receptor inhibitor. So it inhibits GHMSC mediated neuroprotection, and also anti-BP4 inhibited GHMSC mediated neuroprotection, and also wartamanine. Wartamanine is a PI3-AKT inhibitor. So this is also abolished IGF-BP4 GHMSC mediated neuroprotection. So that means IGF-1 receptor and the downstream PI3-AKT pathway are important for neuroprotection by IGF-BP4 in hypoxia damaged primary cortical neurons. So, and also we looked at the phosphorylation of AKT, and we found out that the phosphorylated AKT levels were significantly enhanced in GHMS glial-like MSC treated cells for 30 minutes, and also this anti-BP4 and also PPP actually blocked the GHMSC mediated AKT activation. And also AKT GHMSC treatment actually inhibited Bax translocation induced by apoptosis, because apoptosis actually translocates cytoplasmic Bax into the mitochondria. So we see that here GHMSC actually inhibited Bax translocation induced by apoptosis, but this effect is abolished by anti-BP4, wartamanine, and PPP. So this means that AKT activation induced by IGF-BP4 released from GHMSCs promotes neuronal survival by inhibiting endogenous Bax translocation from the cytoplasm to the mitochondria. And also we as I said before, IGF-BP4 binds to free IGF and stabilizes the IGF, but PA-PPA cleaves this IGF-BP4 complex and to increase the local concentration of IGF on the target cells. We found that the PA-PPA expression is also increased in our glial-like cells, and also glial-like cells actually have increased levels of IGF-1 and IGF-2. So this means that IGF-4, which highly expressed in glial-like cells, plays a crucial role in modulation of extracellular IGF-1 and IGF-2 levels during neuronal protection. So that means these IGF-1 and IGF-2 actually can enhance cell survival in the injured tissues. This is in vitro data, and I collaborated with Dr. Sung-ho Ko at the Hanyang University School of Medicine to investigate the neuroprotective effects of our glial-like cells in vivo model cells. And so we MCAO, middle cerebral arterial occlusion, and then we transplanted our cells one day after this surgery and looked at the animal behavior up to 4 weeks and did the immunohistochemical analysis and Western blotting. And this is MRI results. This is with yellow dots that indicate the brain infarct volume area. And then we found out that GH-MSCC, glide-like MSCC transplantation decreased the infarct volume 4 weeks after cell injection, but the blocking antibody against the IGF-BP4 abolished this neuroprotective effect. This experiment was done by in blind, so they didn't know which cell is what kind of cell is this. And also functional behavioral assay indicates that glide-like cells actually improved functional recovery in brain ischemia. So lower the score, the higher the performance. But the blocking antibody against the anti-BP4 abolished this neuroprotective effects in both animal behavior tests. And also we looked at the immunohistochemistry and found out that glide-like MSC's treatment actually increased phospho-AKT and BCL-X-long levels involved in neuronal survival, but decreased the levels of Bax, cleaved caspase 3 and cytochrome C. They are both markers of apoptosis. And also the Western blotting showed that glide-like MSCC treatment increased NUN, it's a neuronal marker, and phospho-AKT levels, but decreased Bax and cleaved caspase 9. These are the markers of apoptosis. So this is the summary. So our glide-like MSCs actually increased neuronal survival by stimulating IGF-1 receptor mediated signal transduction inside the cell. And this effect was proved by both in vitro and in vivo work, in vivo assays. And these are the conclusions. So we induced the HMSCs into glide-like cells, especially A2 type of astrocytes, without genetic modification to secrete high amounts of factors such as VEGF, HF, and IGF-BP4. And these cells actually alter the tissue microenvironment through paracrine actions of growth factors to enhance tissue regeneration. And finally, our cells can be used to treat the injured nervous system and ischemic diseases by modulating microenvironment. So this is the summary of development of safe and efficient cell therapy for neurodegenerative disease. So these human bone marrow or adipose-derived mesenchymal stem cells can be induced without genetic modification into glide-like cells. And then these glide-like cells have enhanced paracrine actions and resulting in the microenvironment modulation. So we can have a better regeneration of neural tissue. Thank you very much. And then I'll take the questions from the audience.
Thank you very much for President Chang for very nice results. There is a question from the audience, Professor Sekino.
Yes,
Yes.
Thank you very much for your beautiful and nice work. My question is on the technique to introduce your cells into the body. I see that you injected your cells in the healthy tissue that is close to the brain lesion. Do you have any idea on how to maximize the treatment effect by developing any techniques to introduce your cells?
Oh, thank you very much. This is very important issue, actually, because delivery of cells are very important, especially in treating patients. So we thought about a lot of ways, and this is we used the stereotaxic injection in this study, but in humans we can use Ommaya resolve. We can use repeated injection in the brain, so we can use that. Also, we can use so IV injection, but unfortunately some cells cannot penetrate the brain-blood barrier into the brain. So we have to think about to develop the high efficient cell delivery method. That's another big research project, I think.
Okay, thank you very much.
Thank you.
Thank you. Is there any other question?
So I have a quick question,
Nathan.
Professor Chang.
Yes. Yeah.
So once you induce the glial-like cells, can you be able to like? I'm wondering like mass production of the cells and all the conditioned media itself. I think my belief is that the conditioned media can be probably utilized as a therapeutic agent. You know, there's some companies in US they're actually injecting the conditioned media from stem cells for the anti-inflammatory or the disease treatment,
Right.
and I'm sure this can be applied as well. Can you be able to tell me whether these glial-like cells are expendable, and whether you can have a large production of these cell population?
Yes, that's one of the advantages of glial-like cells. It can be expandable.
And
I
do
can
they
have,
maintain their phenotype, like
Yes.
secretion of these? Okay.
yes.
Okay.
So, and also I can freeze those cells in the middle of induction. So we can use whenever we want to, we need them. That's the advantage. So we optimize the condition of freezing and thawing, and also culture method to have enough cells.
Okay. And did you also perform the proteomic? Sorry, just how much is the IGFP4 is actually secreted by the cells? Were you able to compare to other, like is it dominant factor secreted by the cell, or is it glial-like cell, or is it one of the factors?
One of the factors, actually.
Okay.
That's so I think I can use these glial-like cells as your kind of a cellular vector with cocktails of a lot of good stuff. And one of them is IGFP4. And this is, I showed before, but if I just showed it here. In our cells, they secrete around 8 nanogram per ml.
Okay.
But it produced a similar neuroprotective effect compared to 200 nanogram per ml of recombinant protein. So this is very efficient because, as you said before, it has a proper three-dimensional structure and also post-translational modification. So this is much more stable compared to recombinant protein and much cheaper.
Thank you. Thank you very much, Professor Chang.
Okay.
Thank you.
Thank you very much. Okay.
Let's move to the next presenter, Professor Keiichi Nakagawa.
Yes. Can you hear me?
Yes, I can hear you.
Okay.
So the title of Professor